15 minutes each for the lead lectures and 10 minutes each for the oral presentation. So I think that should be okay for the speakers.
Yeah, that's right.
I think that we shall start with Dr. Sridhar. Sir, please, sir.
Yeah, please. The screen is not opening. Mr. Chairman, Mr. Co-Chairman, the IPS South Zone President, Dr. Nallathambi, my friends and associate scientists from the headquarters, and my fellow plant pathologists. During this slot, I wish to deal with some aspects of functional genomics related to disease resistance. This would emphasize the importance of learning about the resistance gene functions that is essential for developing novel crop varieties involving gene modifications for modulating the recognition and specificity between the pathogen and the host to our advantage that might help to check the losses caused by the devastating diseases. I have chosen a set of three resistant genes to deal at the end. So let me share the screen, and while sharing the screen, to keep the conversation on, a little bit on molecular biology. Molecular biology emerged during the 1930s.
Sir, could you share your?
Yeah, it's coming. It's coming. It's coming.
Yeah.
Between a physicist and a chemist. Chemists joined together initially. Physicists and chemists joined together initially to understand the life in principle. Then later it was joined by the biochemists, geneticists, and other subjects. This was named as molecular biology only in 1938 by Warren Weaver, who belongs to the Rockefeller Foundation, who was heading the Natural Sciences Division of Rockefeller Foundation. And probably I think after introducing the subject, we will have a small travel through the molecular biology towards the end with the examples of the three genes which I've chosen. Dr. A.K. Singh and Dr. T.S. Sharma probably, I think, laid the foundation that is necessary for this through their excellent work which is being done at IRA. I think the screen is visible, slide is visible for you clearly.
Yes, sir, it's visible.
Yeah, yes.
Okay. As far as the deployment of resistant genes, for beneficial in agriculture, it has been in use for decades, even though their effects have not always been durable. The deployment of resistance gene, the deployed resistance genes used to break down because of the problem by the strain variations and the mutations in the pathogens. The choice of using a single dominant genes in the breeding programs has been preferred by the plant breeders because it's easy to handle them. It's easy to travel, to transmit it to the new varieties, and also it is easy to study the genetics of it and all those things. So the preference was always to use the dominant resistant genes for developing newer varieties. There was a mention about Floor's gene for gene concept. You can see Dr. Floor here, those who have not seen his photograph. And his concept of gene for gene resistance has come out through 40 years of research in the host plant and the pathogen linseed rust that has taken place in that. It's not all of a sudden the concept has not come up. It took 40 years for Dr. Floor to arrive at the concept of this gene. At that time, the molecular biology was not known. And there was no indication that we can do a lot of biology advancements in biological sciences. But still, if you read the paper, the original papers of Dr. Flo, he visualized, he's a visionary. He visualized that at the end, when he concludes that the host and the pathogen interaction will be more thrown out only when through the molecular aspect of their functions, the function of a virulence gene and the function of the resistant gene from the host. And according to his concept, you know very well that if the A virulence gene and the virulence gene are compatible, then you have the resistance. Incompatible, you have the resistance. When it is compatible, you have the susceptibility. But the Darwin's theory of evolution signifies a threat for survival of the fittest. Always the fittest survives in the nature. And this causes the arms race between the host and the pathogen. Either the host feels that it is safe, nor the pathogen feels it is safe. So each one of them, they try to adjust and then evolve to withstand the stress or tension that is created by the newer strains or modifications in the virulence chain. And essentially, this arms race is only the problem with the agriculture, as it causes the devastating diseases. Whomever is winning, the pathogen wins almost in a short period of time, but it takes a little bit more longer time for the host to come out with the appropriate resistant genes. And now with the advancement of molecular biology, the incorporation or putting together, pyramiding the resistant genes has become a common thing. I think we were the first in 1980s and 90s to bring the technology of marker breeding, marker assisted selection from the International Rice Research Institute. And we produced several gene combinations or gene pyramids in the IR64 and Swarna, very popular rice variety, with the resistant genes conferring resistance to bacterial blight pathogens, Xanthomonas oryzae, Pathovar. And here you see, this is my own experimental field at the National Rice Research Institute, formerly CRRI, somewhere around 2002. You see in the foreground the susceptible Czech variety, and in the background you see all these are gene pyramid, various gene pyramided lines which are planted in the field. And this is with reference to Swarna, all surrounded by all susceptible, and the Swarna pyramided lines is there. However, it is based on the two presumptions. One, the individual genes, to overtake that individual genes, the pathogen has to mutate to all the gene, 2 or 3 genes which are present. For each gene, to the virulence gene has to mutate, and then it takes longer time for the mutation to occur, and then they knock down the resistant genes together. That is one. Secondly, a single pathogen might accumulate the mutations together for all the resistant genes which are available in the pyramidal genes. That also will take time. But so far, this aspect has not been proved with the experimental evidences. But these are the assumptions, and it's fine. People are pyramiding the genes, and then the gene pyramidal lines, are satisfactorily resistant in the fields, and several varieties have been developed. Dr. A.K. Singh has developed astonishingly very well utilized this technology in developing basmati lines, and at the Indian Rice Research Institute and Central Rice Research Institute, several lines which are resistant to blast as well as to blight, bacterial blight. These gene combinations have been prepared, and they are all in the farmer's field. But when you try to evaluate, this is my own evaluations done sometime back when in Cuttack, that is before 2003. The single genes, they are not consistently resistant in all the locations. They are all the hotspots. These are all the various hotspots of bacterial blight. I show the examples of bacterial blight. But the gene combinations are resistant in many of the locations. But again, it depends upon the which gene. Certain gene combinations in 1 or 2 places, they are susceptible, whereas even 3 or 4 gene combinations as you go down the table, they are also susceptible in this. This is for with reference to Xanthomonas oryzae, patho or oryzae, causing bacterial leaf blight. Same is true. The same pattern occurs with reference to blast disease also, caused by Maganaporthi oryzae or Pyricularia oryzae. And the same situation arises. That means when we compare with the assumptions based on which we develop the genes, compared to single gene bearing resistant varieties, the gene combinations also, you can call them as only race specific resistance. It is not non-race specific. That means there are in certain locations, even there are pathogens already available with mutations or with the avirulus gene, which can overcome the resistant genes and may be able to, in fact, see these combinations where they are susceptible, where it's shown in resistance. So this shows that we should not be complacent with the marker assisted breeding technology through which we are now experiencing the successes, that there is always a possibility that even these gene combinations or gene pyramids might break down to disease at some time or other in the future. It may take a little longer, but they are also vulnerable for disease problem. So the successful development leading to the deployment of pest-resistant genes till now has been dependent mainly on genetic approaches using the genes. Here, I would like to introduce one certain things. Apart from the dominant genes, there are a set of recessive genes. In most of the diseases, you may find that the resistant genes are somewhat better even than the dominant genes in their vulnerability for breakdown of that. That's a general experience. So people are trying to use the resistant genes for quite a long time onwards. So this kindly have it in your mind so that we will see an example of the recessive gene. But the holistic understanding, if you understand how the gene functions, what is the function of the resistant genes in the host, through it should be understood through biochemical means at a molecular level, as well as at cellular biological level. All these three combined together, you may be able to know how the resistant gene functions or offers protection against the pathogen. That is more essential. And a lot of studies are now being undertaken, and a lot of information are accumulating. Now, the theory of immunity has been adopted from human beings to plant. We call it as a plant immunity. Now, if you look at the era of 1960s and 70s, relating to Gauman's book or Goodman's book or Horsfall and Diamond's book and all these things, we never accepted the use of immunity in plant because we thought that the immunity is present only in the living organisms where you got a circulatory system where the antigens and antibodies, so they all take place. But Janeway from USA, he invented the plant innate immunity. It's also applicable for human diseases. So there are two aspects of innate immunity. One is called effector triggered, pattern triggered immunity. That is the pathogen, some of the molecules which are present in the pathogen, maybe it's in the outer cell, which are recognized by the host, and the host gene responds to that chemical structure and then responds a resistance reaction. But the second layer of resistance is effector triggered immunity. That's called ETI, or the former one is known as PTI, ETI. And ETI is more effective where a protein or now any other molecule, even including microRNA, is also secreted in polysaccharides, microRNAs, various types of compounds or effectors. They are secreted by the pathogen through a nano machine, which the bacterial pathogens and the fungal pathogens through the other mechanisms inject into the host cells, and these molecules, they interact with the resistant gene protein, and then the response complexity of reference, that's called the recognition and specificity involved in this biochemically or at molecular level. So this might lead us to biotechnological approaches if you understand what is the substance that is being as effector. Morning, I think Dr. T.R. Sharma has made a mention about the effector biology. Now, this is a well advanced field. And what is the type of effector and how that protein-protein interaction takes place, or the protein DNA complex is formed. And then that response is through the cellular changes, give the response that or activates the defense pathway through the hormonal, which are all on the downstream hypothesis. So this might lead to, so these genes are responsible for resistance, we know, but unless we know the functions of that, then it may be difficult to devise where to lay our hands to modify them or for genetic engineering. But at the same time, we should also say that the crop production is determined by the plant architecture, the plant physiology, or the anatomical features of the plant, or the cellular changes that is taking place. Say, for example, increased photosynthesis or reduced photorespiration or anything like that. You know, all these are complicated mechanism that governs the crop productivity. But now, when you are dealing with the resistant genes, you should always have in mind whether your resistant gene or the interaction between the and the gene has got, you know, in the earlier years, we used to say that if you breed resistant varieties, your yield will depress. But that's not the case, what we have seen in the early days. But at the same time, we will take up 3 examples. And these 3 examples, I have taken these, which the genes which will express pleiotrophy. Pleiotrophy, you know, is a character, is a genetic character where a single gene governs more than one trait. You know, we have read that during Jacob Monod's the protein chemistry, that a single gene produces a single protein, and that single protein has got a definite specific activity. But later, the single gene governing 3 different traits, they may not be related. They may be related or they need not be. They may be unrelated traits in the plant system. That is why I say that these unrelated traits might govern some other aspects of your crop production mechanism. You know, that you please keep it in mind. So this example is known, and this is not a rare thing. It is always occurs, but the only thing is in nature that we have to find out which genes they express this, so that can be exploited without affecting the crop yield when we are trying to handle the resistance through the modern technology such as genetic engineering or gene modifications. The first example which I would take is XA13. This was also mentioned by Dr. A.K. Singh. A.K. Singh has used XA13 in his gene combinations. We have also used about it in Basmati. Very impressive. The resistance, this recessive XA13 governs the resistance to bacterial leaf blight. But now we know that the dominant form of XA13 is the one which governs the sugar transportation in the plants. Now, the susceptible varieties they bear the dominant gene, whereas the XA13 recessive gene offers resistance. This XA13 is a natural mutation. Nobody has made any genetic engineering here. You know, it's a natural mutation of XA13 and XA13 recessive, and XA13 recessive gene gives you protection in that. But unfortunately, or fortunately, this XA13 dominant gene as a sugar transporter, this is expressed not only in the foliage, but also in the floral parts. You know, as you see here in the floral parts, the sugar transportation, and this is essential for pollen development. Suppose now, let us take, forgetting about the natural mutation, if you want to genetically handle XA13 susceptible, this is called a susceptibility gene. The susceptibility gene, when you want to modify it and then mutate it in the laboratory and then use it for resistance, you know, it loses the activity of making available sugars for pollens to develop. That means your grain formation will be affected and your yield of the plant ultimately will get reduced. But since it is a natural mutation that has taken place in nature, now this XA13 recessive gene has maintained both the sugar transportation mechanism as well as resistance to bacterial leaf blight. We still do not know what is the mechanism through which the XA13, I mean the cellular mechanism by which the XA13 governs, provides the resistance. But still, we know it only as that XA13 is a good gene for providing resistance to bacterial leaf blight. You can see here, this is also one of my plot at CRRI. This is a susceptible row, check row, and this line carries the XA13 alone. And this XA13, the person who is standing there is an experienced field assistant. I mean, we owe a lot for through his hard work for laying out the fields and other things. We salute such technical persons indeed. So we know that this XA13 is otherwise known as OS8N3 or OS Sweet 11. You know, the rice plant has got, Dr. Sharma mentioned in the morning, about 20 sugar transporters. They may act in different tissues. But one thing is to be free from restricting the sugar transportation to the pollen grain, the gene should express both in floral plots as well as in the foliages also. In both the types of tissues, the gene should express. XA13, the recessive gene, the natural mutant, expresses in both the floral parts as well as in the leaves. You know, the susceptibility gene, you know, it's Johnson's hypothesis which has come out. Even when we were working towards the deployment of resistant genes, I used to discuss with my breeders who are associated with that, what is a susceptibility gene? The breeder used to say the susceptibility gene is nothing but, you know, your resistant genes get mutated and some sequence, there is a chain, some deletion or insertion or something like that that happens. And that becomes a susceptible allele. We call it a susceptible allele. But now we know that distinct susceptibility genes exist. And this is one example of XA13. And let us see, because if the XA13, when it is modified to XA13 in laboratory conditions, you know, it has got some off-target effects. Actually, Dr. Ma and Gao et al., the Chinese scientists, modified the dominant XA13. They wanted to produce another effective XA13 resistant genes. When they modified gene modifications, when it was done using RNA silence and other things, you know, all the technology that are available. But when the plants that has come out of that, they showed off-target effects or knocking out of XA13 enhances resistance.
Sir, we have completed 20 minutes, sir. Now 20 minutes passed from your lecture. Can you?
Oh,
yeah.
You want summary? Okay. Yeah. This is the thing, you know, off-target effects which are coming there.
But the time constraint.
And then the second gene, this is the gene, and the gene modification that has taken place in the promoter region of the gene. So therefore, modifying the promoter without affecting the coding sequence is one possibility that is affected. The next example is OS5 and OS5. This gene governs, you know, resistance to bacterial leaf blight, at the same time tolerance to cadmium stress. these two traits are governed by these genes, and this also, when modifications was taken place, off-target effects were noted. Then twin benefits of a single gene. Another third example is XA4 gene. XA4 in the dominant form offers, it was a very good gene, and later because of development of several races that can attack XA4 combination, XA4 gene alone, it was considered as a defeating gene. But today we know that XA4 offers through the cell wall mechanism and other things, it gives you a good plant type. But still, XA4 acts as a QTL in protecting the plant. To some extent, some protection is given there. So the practice now is to use, it will be a good practice when you want to breed a variety or when you want to marker-assisted breeding, amalgamate several genes together to produce one. It's always better to use XA4 along with the other genes so that your plant type also is good, apart from providing small resistance, and resistance can be added by other genes which you are adding it up. This is another approach which is there into this. So the gene functions, unless we know the gene functions, this type of intrusion or our modifications will result or end up in off-target effects. And there are ways to check the off-target effects by genotyping for various traits, various genes that govern the productivity of crop plants and other things, and how to avoid also genetic mechanisms are available. Here I would like to recall a favorite remark by Professor Swaminathan. Please read it yourself. The days are advancing very fast. Technology is developing very fast. Even if you want to stay in one place, the same place where you are staying in your research, you have to run twice as fast as you could to stay in your own place. But if you want to make an advancement, you can decide how much speed you would like to gain in your running against the technologies and use of these technologies. Well, this is the end of it, as the time doesn't permit to explain it a little bit more. I thank the organizers for their opportunity to talk to you this afternoon. Thank you, and thank you all for this visit.
Thank you, sir, for the wonderful lecture, sir.
If there are any questions, I'm prepared to take it up. Thank you.
Dr. Sivasamy. Dr. Sivasamy.
Yeah, it's a good. Then we can go for next presenter, and then we can ask questions in the last.
Okay. Thank you, sir. Thank you, Dr.
Okay. Thank you very much.
We will take up next presentation by Dr. Ramesh Sundar. So he will be talking on unlocking molecular toolbox of the enigmatic interaction involving sugarcane and smart pathogen. Probably many of you know Dr. Ramesh Sundar as one of the leading molecular plant pathologists. So he will enlighten us on the ongoing activity at our institute on the host pathogen interaction in sugarcane and smart pathogen. Dr. Ramesh Sundar, please.
Respected Chairman, Co-Chair, reporters, and dear fellow delegates, good afternoon, one and all. In fact, I take this opportunity to thank the organizers, specifically Dr. Nalathambi, for giving this opportunity to interact with the delegates of IPS South Zone. And the topic of my presentation is unlocking molecular toolbox of the enigmatic interaction involving sugarcane and the smart pathogen Sporosporium citamedium. In fact, you would agree with the point that there is a paradigm shift.
Ramesh,
yes.
you slightly adjust your camera so that your face will come completely.
Is that okay?
Slightly make it down, yeah.
Okay.
Either you raise up your, the.
Yeah. Now it's okay, Ramesh.
Okay, sir. I will proceed.
Yeah.
So you all will agree that there is a paradigm shift in understanding plant disease resistance, thanks to the emergence of robust platforms of molecular biology, which has really made us to understand the intricate interaction far better than where from we have transgressed from the floors gene for gene hypothesis to what we are now. In fact, plant pathogen interaction is still enigmatic, because it involves two dynamically evolving biological entities, namely the plant and the pathogen. And deciphering plant disease resistance at the molecular level is the foremost step in achieving durable disease resistance. So coming to the topic of our interest, that is SMUT, disease of sugarcane, which is gaining lots of importance in the recent decade. In fact, I would say this disease is a disease of global prevalence, unlike red rot of sugarcane, which is the foremost disease in Indian subcontinent situations. And interestingly, if you look at the pathogenic lifestyle of the SMUT pathogen, this is a typical biotrophic pathogen which has a very tricky lifestyle, wherein which it tries to hijack the metabolism of the host and then makes it more convenient for the survival of the pathogen. Also, interestingly, we could found a typical lifestyle transition of the pathogen, wherein which it has a transition from a non-pathogenic haploid sporidial stage to a pathogenic dikaryotic mycelial stage. So lots of things are hidden or are to be answered from our basic research combined with the molecular approaches. In fact, there exists a molecular switch which exactly regulates the transition of a non-infective phase of the pathogen to an infective phase. So to resolve many such intriguing questions, we attempted to unravel the mystery by resorting to two approaches in our experiments. One would be the pathogenomics, which is nothing but the whole transcriptome analysis of the pathogen, not only the pathogen per se, but also alongside with its interaction with sugarcane. And then we wanted to look into the secretome part of the pathogen to decode the effectors of our interest. And the final part will be, in fact, I would not call it as a conclusion or where we are now and what we would look for the future. And now, interestingly, we have been hearing lots of updates on the understanding of plant disease resistance, right from the inaugural session by our DDG, by Director IARI, by President IPS, and of course, by the senior colleague, Dr. Sridhar, who was just the speaker, who just enlightened us with the updates. See, crosstalk is a very important component, particularly when the plant and the pathogen interacts, because during the course of coevolution, the pathogen has its own metabolites or its own mechanisms by which it can surpass or circumvent the host resistance. To name a few are the effectors, the PAMPs, and so on and so forth. Parallelly, the hosts also, during the course of interaction, will have to put forth its defense very actively to address to the pathogen ingress. So we can call them as the receptors, the membrane-bound receptors, which are nothing but the cognate receptors, which are interacting at the protein-protein level with the effector proteins, molecules from the pathogen side. And of course, these interaction decide the outcome, maybe a resistant interaction or an incompatible interaction or a compatible interaction or susceptible interaction. In fact, these are the three major outcomes which forms part of the interaction process during the host pathogen interaction. One is an effector triggered susceptibility, maybe an effector triggered immunity, that's what we call as a ETA response, or a PAM triggered immunity, what is we call as PTI. In fact, ETA is a very robust response, which normally we have to look in for, and that is what we envisage to have to engineer a broad spectrum resistance. Well, the application of genomics and transcriptomics have definitely revolutionized our understanding on host pathogen interaction. And of course, I would say that proteomics has ably complemented the understanding. And of course, over the recent years, maybe after 2000, we have been seeing lots of whole genome data coming out of many, not only model systems like Aspergil, the model systems. We have lots of genome sequencing projects coming out with the whole genome information of croplands like rice, and of course, sorghum is also in the offing. But parallelly, you could see lots of pathogen genomes have been found to be decoded, and this has added more strength in understanding the plant pathogen interaction. In fact, we have to look in for a very holistic way of integrating all these tools of omics to make our understanding better. During the recent years, we could see lots of new terminologies like phytopathoproteomics, proteogenomics, effectoromics, interactomics, and many such omics platforms, which helps us to critically go and specifically look in for the finer details, which would help us to fill the gaps in understanding. Well, this just gives a schematic representation of the properties of the fungal effector proteins, where we are trying to work upon to understand it better, to understand the role of effectors during the plant pathogen interaction in our context, maybe sugarcane red dot pathogen interaction or sugarcane smut pathogen interaction. These are some of the critical points, some signatures of the effectors, like they are normally small in size. Generally, they are defined as high cysteine proteins, with a characteristic signal peptide and an extracellular protection, the cleavage signal in between. And of course, more interestingly, effectors are the candidates which have to be looked in for from an evolutionary perspective, because effectors are the genes which undergo lots of selection pressure, because the earlier speaker was referring to the breakdown of resistance, possibly due to emergence of new pathotypes, which I attribute to a selection pressure undergoing at the gene level, and that is the effector genes undergo selection pressure and then gets mutated into a different form, which can circumvent the plant defense and then break down host resistance. In fact, the localization of the effectors, mostly the effectors are localized in the apoplastic spaces. The apoplastic space is a typical war zone where the plant and the pathogen put forth its combat system at the molecular level, and then to fight out each other to gain an advantage over the other partner. Of course, there are also elicitors which are delivered to the cytoplasm, as well as which are delivered to the nucleus. In fact, nowadays the computational tools, the bioinformatics tools has come in handy, and then lots of prediction platforms are available, which helps us to zero in on the specific effectors, predict them and identify and then functionally annotate them. So coming to the tricky lifestyle of the sugarcane smut pathogen, it has a typical diploid teleospore. You could see the SEM photograph as well as the light microscope photograph. It emerges in the form of a germ tube, gets into a promycelium, and then develops into a haploid sporidia. Here comes our interest, because the haploid sporidia will have two mating types, will have two opposite mating types, what we call as MAT1 and MAT2, or plus and minus, which represent opposite mating types. They fuse, the opposite mating types do fuse, and then these opposite mating types result in emergence of the infective dikaryotic mycelium. In fact, this is the infective entity of the smut pathogen, not like the haploid sporidia, which is a non-pathogenic entity of the pathogen. And then this leads to intracellular colonization, and then it totally ramifies the sugarcane meristem, and then you could see the emergence of a smut whip. So this is a putative model which depicts the BE gene. BE gene is nothing but the this is the core switch, I would say, which triggers the pathogenicity during the fusion of the MAT1 and MAT2, and this leads to, and the fusion of which leads to a heterodimer, the E and W heterodimer, which triggers the pathogenicity of the fusion. Well, we try to just characterize or isolate the MAT1 and MAT2 by way of classical and molecular approaches. You could see clearly here that the colony 2, this is a checkboard experiment which we performed, and then we could clearly identify the opposite mating types. The colony 2 represents a compatible mating type with others, because you could see the fuzzy growth, and the rest of the colonies 1, 3, 4, and 5 represent the opposite mating type. So followed by this, we went on to phenotypically distinguish the isolates. Like over the past 5 years, we have been screening several isolates differing isolated from different geographical locations, and we could clearly distinguish two different phenotypes. One is a virulent pathotype, what we call as SS, Porcine Cytaminium 97009, and another one is an avirulent phenotype. It is from Andhra Pradesh, that is SS. V stands for Uiru 89101. So phenotypically, we have clearly distinguished two different pathotypes, and then we went ahead to specifically, we designed primers for the MAT1 and MAT2, and then these genes have been found to exhibit clear-cut PCR profiles. You could see both the isolates. We could distinguish the MAT1 and the MAT2, and then we proceeded further in the molecular analysis. We went for different types of markers, and among which the SRAP markers gave us an excellent result, which clearly helped us to distinguish we work out the dissimilarity coefficient here. You could see the smallest dissimilarity coefficient existed between the two high virulent pathotypes, namely the SS96007 and the 97009. And also you could see one isolate which we got it from a resistant variety, that is 6806, was found to share the maximum dissimilarity coefficient. And very interestingly, in concordance with the phenotypic results, we could see the high virulent and the low virulent or the less virulent isolates clustered or grouped into two different clusters. Well, also we proceeded with comparing the intense colonization using the two distinct pathotypes, inoculated, challenge inoculating a susceptible variety, smut susceptible variety. You could see a very clear distinction between the intense colonization observed in the high virulent pathotype, whereas you could see very sparse or just negligible traces of colonization observed in the less virulent isolate. And of course, we went to have an absolute quantification of the pathogen biomass by resorting to qPCR, and then you could clearly see that, of course, we did all the groundwork. We optimized the conditions. We developed a standard curve using distinct concentrations of the DNA. And then we could end up in getting a very high level of pathogen titer when we used the high virulent isolate as you compared with that of the low virulent isolate over different time points. So this has given us enough confidence to proceed to the next generation sequencing platform. So we went for a RNA-seq, we went for a whole transcriptome profiling, and this is the experimental design. You could see that we have two sets of experiments. One is in vitro and another is in planta. And of course, we have used two isolates, namely the virulent isolate and the susceptible isolate. This is the experimental design. And this is what we have been doing here. You could see the different flow of the work. We had basic quantitative and qualitative analysis of the RNA extracted from distinct developmental stages of the pathogen, both from the high virulent and the low virulent. We had two sets of samples, in vitro samples, which will have the haploid sporidia and the infective dikaryotic mycelium. These are the two stages which we want to see what is the difference, what lies behind the transition of a non-infective to infective stage. And we have the implanta samples at different distinct time intervals, each representing a lifestyle, particular lifestyle of the life stages of the pathogen. And of course, we ensured good quality of the RNA, and then we proceeded for the transcriptome sequencing, which we got it outsourced. And this is the bioinformatics pipeline. We have two sets of samples. The first set in vitro sample had 5 samples, and the second set we had 9 samples, put together 14 samples into 3 replications. That was the total samples we got sequenced. And of course, when we refer to the in vitro samples, we have a reference sequence of the Sporicerium citaminium published from a South African group, and which we used here for the alignment. Of course, sugarcane, you know very well that the whole genome is yet to be deciphered. So we are helpless by having a whole genome transcript for alignment. So we just made use of the cDNA sequences and then try to make some meaningful alignments. And this is the outline of the transcriptome data analysis part. You can see that the yeah.
Can you please conclude?
Yes, sir. Yes.
Yeah. Yeah, please.
So this is just the outline of the transcriptome work, and it's more of a bioinformatics pipeline, which involves lots of computational biology tools, which will make us to make sense out of the data which we have generated. And this is the raw read summary. You could see we have got more than 300 GB of raw reads. And of course, we made all the quality checks, and the past ones are having either equal to or more than 30 Phred score. And of course, the analysis is still in progress, and then we will maybe shortly in another 3 to 6 months time, we'll be able to come out with a very clear picture. And of course, we did the DGEs. What we know is the differential gene expression between pairwise combinations. And then this is the summary of the results in our pairwise differential gene expression results. And this is nothing but a volcano plot which expresses the upregulated ones, the red ones, and the downregulated ones with the neutral ones, and which also gives a statistical compliance. And this is the heat map analysis profile of the DGE database data set we have developed. And of course, finally, to end up with the functional annotation, these are the results which clearly gives us a representation of a dominant class of hydrolysis. And of course, the functional categories are put into 3 biological process, molecular function, and cellular components. And of course, we used lots of computational biology tools to analyze the data, the huge chunk of data what we have got. And this is the second part of the work which I will just run through, because there will be another presentation by one of my colleagues. To be very brief, here we just wanted to establish a GFP tagged Sporothorium pseudominium for making the histopathological analysis more visible, so that we can understand what is happening between pathotypes of differing in virulence. And this is the success of isolating the protoplast, the first of its kind. We have got it a very convincing result. This is a shuttle vector which we got it from a collaborator from NIST in Trivandrum. And this shuttle vector, we used it for the PEG-mediated transformation protocol. And you could see the positive clones coming up in hygromycin selection, because hygromycin is a selection. And then you could see the GFPs, which are fluoresced only in the case of the transformed cells, and you are not seeing them in the untransformed ones. Here you could see the conformation of the fusion of the transformants. You could see a very positive plus and minus haploids getting fused, and then they will lead to the formation of a promycelium. Then we went on for confirming the integration of the GFP gene as well as the hygromycin gene. We have got very positive PCR results for both the genes, the targeted genes, the GFP and hygromycin. And this is the last part of the presentation. We went on to mine the secretome part of the Sporothorium pseudominium. And to sum up, we finally ended up, after passing all different stages of screening or filtering using different bioinformatics platforms, we ended up in identification of around 82 candidate secreted effector proteins. So to conclude, we are trying to integrate the different tools of the omics platforms, so that we will end up with a holistic understanding of the role of effectors in sugarcane smut pathogen interaction. And of course, the expression pattern of all the transcripts during the crucial infection stage, which would help us to understand better about the dimorphic transition, or what is happening between the transition from a non-infective haploid sporidia to an infective dikaryotic mycelium. And this way we could identify the molecular switches that are responsible in turning on the virulence of the smut pathogen during its infection of the sugarcane meristem. And finally, we dream to end up with engineering the effector triggered immunity response in sugarcane against the smut pathogen. These are snapshots of some of the publications of our group. And I thank all our research scholars who are very much a part of all the interesting results we have generated. And thanks for all ICR, DST, DBT, and the International Plant Proteomics Organization, which have altogether have supported very well in our endeavor to undertake this research. Thank you once again.
Thank you, Dr. Ramesh. So we will move on to next presenter, Dr. Jahirdar from US Dharwad. So he will be again, there's another paper on host resistance. So this paper will be on advances in breeding for resistance against crop diseases, with a special reference to soybean varieties in India. Dr. Jahirdar.
Sir, if he is not available, we can proceed to the next presenter. Let us not waste the time, because
okay.
they know very well that they have to be here.
Okay, Dr. Thambi. Then we will take up oral presentation and we will have a discussion at the end of all the presentation. So now I will call upon Miss Lakshana from our own lab. Miss
Yes,
Lakshana.
sir. Yes, sir.
Yeah, she is our young and dynamic research worker. So working with Dr. Ramesh Sundar. So she will be presenting on the PAMs and effectors coding for red rot pathogen. So Lakshana, please, you confine your talk within 10 minutes, please.
Yes, sir. Thank you, sir.
You will be
Yes, sir. So good afternoon, chairman, co-chairman, rapporteurs, and everybody join with me. Today I'm here to present on side-acted mutagenesis in PAMs, effector coding genes of Coltrotrichum falcatum through PEG-mediated protoplast transformation system. Red rot disease of sugarcane, which is caused by the heavy biotrophic fungal pathogen, Coltrotrichum falcatum, is a serious threat to sugarcane production in India. So there is a pertinent need for identification of an effective eco-friendly compound that can suppress disease either by targeting C. falcatum or by inducing innate immunity in sugarcane. So briefly, when during plant pathogen interaction, plant identifies pathogen through the perception of their conserved signatures called pathogen-associated molecular patterns and trigger pathogen PAM-triggered immunity. Pathogens evade PTI by secretion of effector molecules, and in turn, plant employ R proteins to recognize and activate the effector-triggered immunity. So these PAMs and effectors, they can act as resistance inducers, and they can trigger active resistance in host. So this mechanism is called as the pathogen-derived induced resistance. So Ashley et al. From a research group have identified two pathogen signatures, namely EPL1, eliciting plant response like protein 1, and PDIP1, plant defense inducing protein 1, as a putative PAM and effector that have the potential to trigger innate immunity in sugarcane. So to decipher the functional role of the putatively identified PAM and effector of C-falcatum, site-directed mutagenesis in EPL1 and PDIP1 in trichotrichum is essential. So to accomplish this, the following objectives were framed, namely the standardization of protocol for protoplast isolation from germinating conidia and to establish PEG-mediated protoplast transformation system. Later, to design and construct mutagenesis vector for generation of gene mutants by homologous recombination, thereby developing CF-EPL1 and PDIP1 mutants by PEG-mediated protoplast transformation system. So this is the experimental outline showcasing the standardization of protoplast isolation, thereby designing and construction of mutagenesis vector, and to screen the fungal mutants for hygromycin resistance. So hitherto there is no standardized protocol for protoplast isolation in this multinucleated fungus. So we develop a novel protocol for protoplast isolation from the germinating conidia using lysine enzyme and glucanases, and thereby establish PEG-mediated transformation using PASP vector. So you can see that the beautiful globular protoplast that lack the cell wall doesn't take up the chitin-specific stain, chalcofluor, and doesn't fluoresce under UV, whereas the control does. So this protoplast was transformed using PSP. Here are the transformants. Here are the transformants obtained by transformation using circular vector and linearized vector. Further, since the vector is tagged with GFP, the transformants exhibit green fluorescence, namely the mycelia, conidiophores, conidia, everything exhibits green fluorescence under UV, whereas the control doesn't. The transformants were confirmed at molecular level by PCR targeting hygromycin and eGFP. Hence, the transformants, these GFP transformants can be used for later studies. Then, gene deletion mutants were planned to be developed by replacing the ORF of gene by hygromycin resistant gene by homologous recombination. For this, we construct a novel mutagenesis vector that contains 1 kb of upstream flanking region, followed by hygromycin capsid, and 1 kb of downstream flanking region, in between the HIN3 and the segment of PUC19 vector backbone. The hygromycin capsid sequence was fetched from PUC ATPH vector. So once upon transformation of this mutagenesis vector into C-falcatum protoplast, the gene of interest gets replaced by hygromycin resistant gene by homologous recombination. And this will generate gene mutants which is resistant to hygromycin selection. So these are the vector map of the designed mutagenesis vector for EPL1, for generating EPL1 mutants and PDAP1 mutants. The mutagenesis vector was synthesized by GenScript. Later, it was transformed into E. coli DHP alpha competent cells, and the colonies were screened by vector PCR for hygromycin and for gene-specific upstream flanking region and downstream flanking region sequences. And the colonies were confirmed by restriction and digestion using HIN3 and SAC12. Further, to generate EPL1 mutants, the vector PUC mut EPL1 was transformed into C-falcatum protoplast, and ABC are the transformation controls, whereas the D and E are the two putative gene mutants. So further, The genomic integration of the transgene was confirmed by PCR for hygromycin. And next, to assess the homologous recombination event, we used PCR targeting upstream flanking and downstream flanking region. And interestingly, the colony showed characteristic amplification for homologous event as well as for non-homologous event, as in case of the wild type, depicting the pathogen, depicting the mutants to be a partial gene mutant. So hence, these partial gene mutant cannot be used to characterize loss or gain of function. Hence, experiments have to be conducted again to generate homokaryotic lines that with complete gene deletion. So to conclude, I wish to say that the novel protocols so developed for protoplast isolation can serve as a model for similar works in other filamentous fungus, and the GFP tagged C. falciparum generated in this study can be used to visualize implantar socio-temporal colonization of the pathogen. Further, the mutagenesis vector generated can be accustomed for generation of mutants by homologous recombination in any filamentous fungus. And futuristic work include protoplast-mediated transformation using the two mutagenesis vector to generate homokaryotic mutants and to carry out bioassays for fungal pathogenicity, then host defense response, then gene expression analysis. Further, the gene generation of CFUPL1 and CFPDAPN mutants would delineate its role in pathogenicity and loss or gain of function in virulence in the pathogen. Thank you for the opportunity.
Thank you, Ms. Lakshana. Thank you for completing the presentation in time.
Yes,
We shall move on to the next speaker, Dr. Jagadish from Mysore. Dr. Jagadish is there? I couldn't see his name in this.
He is trying to join now, so you can proceed to the next speaker.
Okay. Dr. Sainamol Kurian is there? From KAU, Professor Sainamol.
No. These are there. Deepa Shankari.
And whatever Dr. Deepa Shankari,
Let's check. No, she has not joined.
Not joined.
No, Sainamol is not there.
Dr. Bindurai is there. So
Yes, election duty. Yeah.
we will take up.
Yes, she has it.
We will take up a Bindurai's lecture. So she will be talking on virus-induced gene silencing, a potential tool for functional genomics in rubber.
लेकिन वो जल्दी सभी दिया है ना
नहीं जितने भी registered delegates थे ना
हाँ
Hello.
Yeah, go ahead.
Yeah, please go ahead.
Okay, sir. Respected chairman of this session, distinguished delegates, ladies and gentlemen, the topic of my paper is virus-induced gene silencing, a potential tool for functional genomic studies in Hevea brasiliensis. Hevea brasiliensis, the rubber tree, is widely cultivated in Southeast Asia and produces latex of commercial utility. And diseases are a major constraint for higher productivity of natural rubber. So with advances in functional genomics, proteomics, and post-pathogen interaction studies, the molecular mechanisms of disease tolerance is being unraveled, and this is how we achieve efficient disease control. Now, our paper, which appeared in Current Plant Biology recently, used transcriptome sequencing and a considerable number of differentially expressed genes that play major roles in response to pathogen were identified. And we know that differential expression of a gene is an indicator of its functional role. In-plantar functional characterization is necessary for understanding the precise role of these genes in combating biotic stresses. So in-plantar gene functional studies depends on stable genetic transformation, and as far as Hevea brasiliensis tree is concerned, this is recalcitrant to stable genetic transformation. It is very laborious and time consuming because it has a long life cycle and there is low transformation efficiency. So now what is the alternative approach? An alternative approach is virus-induced gene silencing, or the WIGS technology, which is a transient reverse genetics tool for characterizing gene functions, and it has been widely used in many plant species. WIGS is a method that takes advantage of the plant RNAi-mediated natural antiviral defense mechanism. And this is how the WIGS works. In plants infected with unmodified viruses, the mechanism is specifically targeted against the viral genome. However, with virus vectors carrying sequences derived from host genes, the process can be additionally targeted against the corresponding host mRNAs. When a virus infects a plant tissue, the spread systemically throughout the tissue, and the endogenous gene transcripts, which are homologous to the fragment inserted into the viral vector, are degraded by the PTGS. Now, what are the advantages of the WIGS system is the effects can be observed within 3 to 4 weeks' time as compared with the time-consuming stable transformation. It is convenient, rapid, and efficient, and is the best method for gene function studies in non-model species with plants with long life cycle, such as in rubber, which is a perennial tree crop. Now, for this, we make use of reporter genes or marker genes, which are used as internal reference. This is mainly used to visualize the extent of silencing to compensate for the non-uniformity of the virus-induced gene silencing. So in this case, the most commonly used reporter gene is the phytoene desaturase, or the PDS gene, which catalyzes a key step in the carotenoid biosynthesis pathway. The inhibition of the PDS expression results in the photo-bleached spots. Thus, leaves that are silenced turn on, become whitish because of the lack of carotenoids and destruction of chlorophyll by photooxidation. Now, the most commonly used vector for the WIGS studies is the tobacco rattle virus, and it causes relatively mild disease symptoms and often induces intensive and uniform silencing phenotypes. Thus, the TRV, or the tobacco rattle virus, also infect meristematic tissues, which allows silencing of genes in growth points. So this is the tobacco rattle virus, which is a bipartite single-stranded RNA virus with one of the broadest host ranges among plant viruses. Now, the methodology that is adopted is the RNA1 and the RNA2 plasmids were transformed into agrobacterium and used for control infiltration. Similarly, I was mentioning about the PDS gene, or the phytodesaturase gene. The TRV PDS vector was constructed and the agrobacterium was transformed. The agro-infiltration studies, we made use of 3 different techniques. One is the syringe infiltration, the agro-drench, and vacuum infiltration. This was conducted using agrobacterium cultures carrying both RNA1, TRV RNA2 with the PDS fragment. So this is, in a nutshell, the protocol adopted. As in other cases, RNA isolation, followed by cDNA synthesis, PCR amplification of the PDS gene to confirm the incorporation of the PDS gene, cloning of PDS gene into the RNA2, and the agrobacterium transformation, followed by agro-infiltration, followed by optimization of gene silencing, which is a very important aspect. It requires a lot of effort to optimize for gene silencing, and ultimately real-time PCR validation of the silenced phenotype. Here I show you the plants that are established for in vitro studies. So these are the Hevea brasiliensis seedlings established for in vitro studies. And I said we have the TRV1, RNA1 and RNA2. So initially we have to make use, or we have to confirm that the vector is there. So we have confirmed by PCR amplification, TRV RNA1 of 210 base pair and TRV RNA2 of 345 base pair. Then we went in for construction of the vector, WIGS vector. The RNA was isolated, cDNA was synthesized, and then we went in for amplification of the beta-actin, which is the housekeeping gene, and then the amplification of the PDS gene, which clearly indicated that the PDS gene has been incorporated into the fragment of about 340 base pair. Now, after the incorporation, we went in for colony PCR to confirm the transformance with TRB, RNA1, RNA2, and the PDS into agrobacterium. So here we have the RNA1, RNA2 amplified, and the PDS gene which has been amplified. The colony PCR confirmed that it has been incorporated. Now, I said that we have made use of 3 different techniques for agro-infiltration. One was the vacuum infiltration, wherein we grow it on what do you call these, the conical flask, side arm conical flask, and subject to 600 mm pressure, and then vacuum infiltrated. For agro drenching, we drench the vermiculite or the soil substance with the agrobacterium culture. And syringe infiltration, we directly take the syringe with the agrobacterium laden culture and then infiltrate it with using syringe. And once that is done, the RT-PCR confirmation of the TRV replication to know that the viral movement in the newly emerged leaves has taken place. So this has been confirmed by PCR, and we could find that both syringe and vacuum infiltrated leaves showed photo bleaching effect, which lasted for within 3 to 4 weeks post agro infiltration, and which lasted for over 2 months. So this is the picture. You can see this is the empty vector control infiltrated plant, and here it is the PDS silenced plant. As I mentioned, that the PDS silenced plant has clearly indicated that there is photo bleaching, revealing that the PDS has been incorporated. So similarly here also, you can see, I said meristematic tissue, even the newly emerging leaves has shown photo bleached effect in the PDS silenced plant compared to the empty vector control. In vacuum infiltration also, the PDS silence plant clearly showed the photo bleached effect, and even in the here also, it lasted for 2 to 3 months. This plants which lasted for 2 to 3 months continued to show the photo bleached effect. Here, down regulation initiated 3 to 4 weeks time. Within 3 to 4 weeks time, you can see the photo bleached effect is present. Agro drench, I said there was no significant phenotypic or molecular level change observed phenotypically. So the empty vector and the TRV PDS infiltrated plant showed similar phenotypic expression. So finally, we have to go in for PCR, real-time PCR validation to understand the downregulation of the PDS gene expression at the molecular level. And here it is very much evident that both syringe infiltration and vacuum infiltration, there is downregulation of the PDS gene expression. So to conclude, the TRV-based system optimized using PDS endogenous gene as reporter, and downregulation initiated 3 to 4 weeks post-agro infiltration and persisted more than 2 months. And the TRV vector carrying the gene of interest can be syringe agro-infiltrated into the lower side of the leaves. And the optimized protocol can be used for validating the role of genes identified in the genomic profile study. Now, what are the future perspectives of the study? Now, construction of the Wix vector of selected candidate genes, defense genes, which demonstrated the differential gene expression in resistant and susceptible clones of rubber, that is in progress, to understand the defense mechanism, facilitating in developing strategies for priming defense signaling during pathogen infection. Thank you very much.
Thank you, Dr. Bindu. It is wonderful work, and the rubber is a difficult craft for Wix work, but still you have standardized. And
yeah, thank you.
Will have a better future. And let me ask for other delegates, those who are to present. Let me know whether Dr. Jagadish is there. Jagadish, then Dr. Sainamol Kurian and Dr. Deepa Sankari. I think that 3 of them are not there. So we will take up the discussion. Dr. Sivasamy, please.
Yeah. I think other 3 are not coming.
Yeah, I think they have not turned up. That Bindu, please close your
Your screen, sir, your screen is to be taken out. Dr. Bindu?
Yes, sir. Is it okay?
No,
No,
not
it's still.
still it is listed.
the share screen. You should come out from it.
Unscare, you just.
Hello, sir. Hello.
Yes, still it is there. Screen is there.
Yeah.
Is it okay?
Yes.
Yeah.
Yeah, we will take up discussion now, Dr.
Yeah, please. There was an excellent presentation. I first of all thank Symposium Organizing Secretary Dr. Naltambik for giving the opportunity for me, and also the IPS Secretary Dr. Robin Gogi and President Dr. P.K. Chakrabarti. And this topic on the advance in host resistance and disease management is a very relevant topic for the present situation. And particularly the presentation made by lead presenter, Dr. Sridhar, was really excellent, followed by Dr. Ramesh Sundar. It's very excellent work carried out, and all the other presenters also made an excellent presentation. I have some questions to Dr. Sridhar. Dr. Sridhar is around? Yeah. That's up. Actually, you mentioned about the recessive gene and the dominant gene. Actually, they are both are race specific or race non-specific. Recessive, because in wheat we see in a contrast, we have most of them are 99% of the genes so far cataloged of dominant one, and the race non-specific ones also dominant, but they are APR, adult plant resistant genes. But here you could give the difference between the recessive and the dominant. Can you throw light on this, please?
Sir, please unmute, sir.
Sir, please unmute.
Yeah.
Yeah, both XA13, the recessive genes also are race specific. Okay. We have experimented with the XA8, XA27, all are recessive genes. Yeah. They are also race specific. They also get knocked down by certain races.
Yeah.
So right. We don't have any non-race specific resistance so far known in rice. But the only thing is, Dr. Sharma, I was associated with his project as an advisor. In the beginning, that's what he mentioned. You know, he was trying to make the protein fusions of both the resistant gene as well as the AVR protein. The aim was to make a non-race specific resistance, but could not be achieved towards the end, because by the time the scheme got terminated, but we still at it. Yeah. So all these are race specific.
Yeah.
Including the gene pyramids, as I said.
Okay. So do you expect this, you are pyramided 3 or 4 genes, but still the pathogen also mutates? Is there a case that the pathogen mutates the multi-loci?
No, the pyramided genes, when it was evaluated in multi-location trials, in certain locations, they are already susceptible. That means the races which which are compatible with those even pyramided genes are in nature available. So when in nature available, possibly by mutation. So when you are forcing the pathogen to a pyramided gene in a location where there is no, the particular races are absent, definitely the possibility of mutation exists, but it may take a longer time. So the durability will be for more number of years.
Yeah. Then we have the approach pathogen-derived induced resistance. What's the last that Bindu made a presentation, where they could take it to the field level through various methods, various agro techniques, or the vacuum techniques, or the injections and so on. How we are going to do such programs in the grain crop, like cereal crops like rice and wheat, in your opinion? The application part.
The application part of it, by the time you reach the application part of it, you have to make sure that you don't have any off-target effects. So for experimentation, it's fine. For understanding, we need the knowledge. We need the knowledge to be gained. But ultimately, when you want to put it on the varieties and then get into the field, when you enter into the field conditions, so you have to satisfy yourself that you are not losing any other good traits.
Right.
Possible that risk is always there. Well, there are certain success stories where they have used the effector gene transmission and all these things. They call it as even effector breeding and all these things. These possibilities are there. And there are a few cases where they have succeeded in this, and then it's possible to do that. But one has to be more careful with that.
Yes. Thank you, sir. This is an excellent progress, and particularly in this field. So my personal congratulations to each one of you who made the presentations. Thank you
very much.
Any other delegates they would like to interact with our lead presenters and Dr. Bindu?
Yes, Dr. Kumar.
Yeah, I was listening to the talk of Dr. Sridhar and the rest. I have a kind of small suggestion from Sridhar. What is the way to delay the emergence of new pathotypes in agricultural system? Because we need uniform type of crop in large area to feed the so-called agricultural production. So we cannot really bring too much of biodiversity in agricultural fields. One way is origin deployment, and how to delay the emergence of new pathotypes. In your opinion, can you suggest something? In addition to host resistance, what would be the compatible technology which can delay, in particular, the emergence of new pathotypes?
Well, the possibility is only to have as much diversity in your genotypes which you are putting into the field conditions. So you can have the genotype genes which are available in your wild germplasm, bring them together and then bring it into the cultivations. Even when we talk about a variety which is carrying a single gene, we do not know whether it's only a single. Unless you are going to develop isogenic lines, you will not be sure that you are dealing with only one gene. You will be having many other genes which are not known. So as much as possible, if you have got the diversified genotypes, then you can delay the challenges that you are posing to the pathogen to mutate. That's only a possibility.
Yeah, but that could be the long-term predictions and projections. But what I'm asking is, can we combine two different crop protection technologies in addition, instead of depending only on the host resistance? Can we bring some very complementary technologies like biocontrol, or even the safe chemicals, if at all any? They delay the emergence of new pathotypes by interfering the virulence. I think some thinking process has to go in this line, so that the best variety of the breeder is not lost just because one pathogenic variant comes. I think it would be wiser not to depend entirely on the host resistance for crop disease management in future. We need to really integrate.
Sure, it is. Ultimately, you boil down to integrated disease management or pest management altogether. Yeah. So you keep your resistant, whatever the resistant line, whether it is genetically engineered or conventionally bred, you keep it in the center. And wherever it is possible, if you have got a biocontrol agent, if you see any changes in the pathogen populations, you can always combine it. So go for an integrated pest management. That's only a possibility of that. That's always good also, good and safe also. At the same time, we will save the breeders' materials that are available at the moment. We will not throw it out immediately as far as possible. So it is not that host resistance alone is going to provide you the solution. Well, host resistance can give you a central figure to move around how you are going to manage the pathogen populations which are available there. Generate knowledge
Thank you, sir.
about generate knowledge about the pathogen structure that is available in each of your locations. Know what are the type of virulence genes which are available there into that, and accordingly you breed varieties and combine your bio agents and other things for managing, or even cultural practices should be possible.
Yes,
That is a meaningful way. That's a meaningful way.
Can I add to what Dr. Kumar has asked? Just a point.
Yeah, please.
Okay, with the permission of the chair. In fact, there are a good number of reports on exploiting biocontrol agents, which can interfere with a typical example is the sugarcane smut pathogen, where people are trying to make use of biocontrol agents which can smother the mating type genes to fuse. So that will typically switch off the pathogenicity per se. So I think lots of promising results are there.
I do not know whether any molecular evidence is available.
Yes, sir. There is a published paper in sugarcane smut pathogen where they have used biocontrol agents. That's a work done in Brazil and Australian collaboration program. So it's just one paper which I've seen where they have tried to hamper the fusion of the opposite mating types of the smut pathogen. So that led to loss in pathogenicity. So maybe it might open up vistas for the future research.
It's a good concept. It's a good concept. If the mating is possible, it depends upon how the mating takes place. If the mating is possible, you may be able to subdue the virulence population. You are not acting on the virulence gene, but you are only lowering down the virulence population that is available. By managing the population, you can achieve certain things, but not the genes will not interact with that, lowering the virulence genes. That will not get mutated.
Any other delegates would like to interact with Dr. Sridhar or Dr. Ramesh? So since we have other absentees, so we have enough time for a discussion. If not,
I have a question for Dr. Ramesh Sundar sir.
Yeah, yes.
It was interesting to listen to his integrating tools of omics platform and the transcriptome data, sequencing data. Sir, you have identified, listed out several genes. Now, I was just, when we give for outsourcing, we do get a lot of data, lots of upregulated genes, lots of downregulated genes. So it's not only the upregulated genes that we are concentrating on, the downregulated genes. I just want to know how are you going to shortlist and pick the right gene, because we cannot handle all these thousands of genes, or even if we shortlist based on the heat map and all. To what extent do you come down, and how do you shortlist?
In fact, we are in the process of analyzing our whole transcriptome data, and we are halfway through. And rightly said, like we have to depend upon the published information available in this particular case. We have some good reports published from the Australian group and from the Brazilian group. So we are trying to shortlist the most potential ones, which we can look in for promising, which will have a functional relevance in our context. And in fact, in one of my slides, we tried to go in for in silico secretome prediction analysis, where we have identified potential number of CSEPs. In fact, we have cut down the number, and then probably we'll be handling a few good number of genes which we can functionally validate after expressing, after doing the qPCR, doing the expression profiling. Probably we'll go in for the functional annotation. The potential ones, we will go for functional annotations, looking for the gain or loss of function. And then probably, I think that's the way out we can proceed further.
Thanks very much, sir.
Dr. Vishwanathan, I have a small interaction with Dr. Ramesh.
Yeah.
Dr. Ramesh, I'm just curious to know, did you find any effector coding genes in Schistominia genome?
Yeah.
And can you tell what are they actually in terms of chemical, if protein, if it is known?
No, it has not come to that level. Our data analysis part is just, I would say, maybe 60–70% of the, we are just proceeding with the data analysis. Maybe in a couple of months time, maybe 3 to 4 months, we should be able to come out with a very clear picture, the functionally characterized, the ones, the effectors, which we can pull out from the whole transcriptome data set. Yeah, we look forward to pull out some promising ones, then we will proceed further in functionally annotating them. Yeah.
Okay, thank you.
I have a question to Dr. Bindu Rai.
Yes, sir.
You have just given an excellent presentation on VIGS silent methodology. And
Yes.
what are the effect of the diseases which you could able to address? You just mentioned about the disease control holistically, but specific diseases in the rubber plant.
As I said that through the transcriptome study, we have been able to identify certain genes, and now we have standardized this protocol for the virus-induced gene silencing. Protocol is optimized, so now we will be able to incorporate these genes into the PDS along with the PDS, and then proceed with this silencing part. And we have standardized which methodology is also efficient. We have standardized the VIX protocol. We have standardized the methodology which could be used, like the
And what are the diseases you can able to address? Which are the diseases?
The major diseases in rubber are caused by Phytophthora and the Corinospora and Colitotrichum. So we will be, to begin with, we will be starting with the Phytophthora resistance.
A wide range of diseases you can able to.
Pardon me?
Wide range of diseases you can.
Yes, not many diseases, but in case of rubber, it's all fungal diseases, and the three major pathogens are Phytophthora, Corinospora, and Colitotrichum. So to begin with, we'll start with the Phytophthora resistance. Sir.
Okay.
So one small query to Dr. Bindu.
Yes, sir.
Do you have any genuine resistant type of rubber against Phytophthora? Do you have any?
We don't have a resistant variety. Comparatively resistant variety we do have. Tolerant variety we do have, which is a interspecific hybrid of Hevea brasiliensis and Hevea benthamiana, two interspecific hybrids. So we use that as a resistant variety to compare with a susceptible variety.
Okay, then what is your future plans if resistance is not known in the naturally available cultivars? How are you going to incorporate or identify?
Dr. Sridhar sir was telling, we are on the lookout for germplasm varieties to get some really resistant ones. We have a collection of over 4,500 germplasm accessions collected from the wild Brazil. So we have those germplasm accessions with us. Screening is in process to identify. There we are able to get some really very good resistant variety accessions from there. So probably, like what Sridhar sir said, we will be making use of the accessions, germplasm accessions. Although they do not have a good yield, they do possess good amount of disease resistance.
Okay, I believe this Phytophthora causes abnormal leaf fall in rubber.
Yes,
can I add a comment?
I just wanted to, I'm just curious to know, in fact, ever since I was reading this article, this disease, what is its ecological significance, the abnormal leaf fall?
The abnormal leaf fall disease happens along with the monsoon period, Phytophthora, with the rain season. So what happens is the entire canopy, the leaves of the entire canopy, it just falls. So one plant gets affected, it spreads to the entire plantation. And as I said, there is no good tolerant variety. So the high yielding variety is invariably highly susceptible to this disease. So the entire canopy falls, and it leads to disease yield loss of up to 40 to 56%. Because there is no canopy for at least 3 to 4 months. And this happens soon after the wintering period. So the wintering period is sometime during December, and then all the refoliation takes place during February. And
exactly,
all.
I was coming to that point only.
Yeah.
When the leaf fall happens, there must be some benefit for the host as well.
Yes.
I think we need to investigate what the gain for the host, although it's a disease and there is a reduction of rubber yield. But there must be a survival fitness for the rubber plant, mediated by the environmental cues. I think Phytophthora is only one factor here.
Yes, sir.
And what rubber gains out of the whole phenomenon, that we need to study.
Yeah, the rubber gains out of the whole phenomenon is the wintering process, which happens naturally. But the disease causes the abnormal leaf fall disease. So it's the leaf fall which is not desirable for the plant. So it's an abnormal leaf fall that happens. So
Yeah, the
that
plant
leads,
regains, right?
yeah, the plant
The season
regains
changes,
after the
phytochry is
wintering.
no longer
No.
able to survive, then the plant regains.
So as I said, in December, the wintering takes place. By January, February, March, the entire foliage happens. And then the plant is in a really good mood to start assimilating. And at that time, when the infection takes place, the entire canopy falls again. You know, so it's
that sounds
going
good, but
to...
still I have a feeling there must be ecological fitness for the rubber. The whole phenomenon is not worked out. Yeah.
But we have done a lot of studies on the yield loss due to abnormal leaf fall, and then we find that just because of the leaf fall, there is up to 40 to 56% of leaf yield reduction as far as the natural rubber is concerned.
Thank you.
Thank you, sir.
Yes, Dr. Sridhar, sir.
Yeah, I just want to add a point. You know, when we say that the off-target effects, you know, because of the time restriction, the point could not be reached there. Yeah, off-target effect can also be beneficial. Say, for example, when they try to modify the XA30, recessive XA13, the modified XA13 without affecting the sugar transportation is able to provide resistance to sheath blight disease, for which there is no genetic resistance that is available. Resistance means it's a lowering the susceptibility grade of the rice to sheath blight disease. That also is possible in that. This may be in the connection with the discussion that is going on in that. So that should also be possible.
Thank you, sir. Sir, I have one suggestion. I want a suggestion from you. So we are hearing mostly the host plant resistance, successful host plant resistance, exploitation, mostly with regard to rice, wheat, and few other crops where genome or genetics is very clearly known. So we are working in an orphan crop like sugarcane, where this inheritance is not very clearly explained. So, but still we are going on and developing varieties.
Please mute. Please.
So I'd like to have your views on that. Even after 100 years, we are in the same situation. So that the stability of resistance is not there, and regularly we are losing.
There is some disturbance.
One after another. So with your experience, you can suggest us at least where we can look for. At the pathogen side, we are working now. We are working on the effector molecules and other genomics also we have understood. But the host side is very, very vast, complex polyploid.
Okay. I'll tell you. Sir, we have to, it is better to wind up at the earliest because we are waiting for the poster session. People are waiting.
Yeah, that's right. When you are working on.
Yeah. Dr. Sridhar, sir, you would like to tell some? With his comments, we will conclude.
Please.
I think that the session, we are going to conclude the session. So, Dr. Sivasamy, shall I conclude the session?
Yeah, go ahead, please.
Yeah. So that we have that all the presentations, but we had excellent presentations from Dr. Sridhar, Dr. Naladambi, Dr. Ramesh, Dr. Bindu, and also our young researcher, Ms. Lakshana. She also presented very well. And these topics are very relevant, and actually even after a presentation we had enough extensive discussion also. And I think it has opened up this session, opened up new areas to understand and to further proceed on this area of work, especially on the omics tools that Ramesh has explained. So we will move on to the next session. So thank you all for the hearing and also for active participation. And thank you IPS Society for giving us the opportunity. Thank you. First of all, thank you the speakers. So even Dr. Sridhar has done extremely very well in his presentation. Due to time shortage, he could not continue fully. And also I thank Dr. Kumar from Delhi and also from IPS colleagues from the headquarters. Thank you once, one and all. Thank you.
From the organizing side, sir, we are Dr. Sridhar, sir. We are very grateful to you. All the way you could make it possible to attend this symposium. And my colleague Dr. Ramesh and Dr. Bindurai, all the delegates really were very overwhelmed out to your presentations. So thank you very much. We extend all our wholehearted thanks to all of them. Thank you. And I also thank Dr. Viswanathan and our head, Dr. Sivasamy, for nicely conducting the session, though there were very few presenters, and of course the rapporteurs and all. Thank you, sir. Thank you, one and all. Thank you very much.
Thank our rapporteurs also. I forgot to mention Dr. Anjuman.
We are thankful to you. Thankful to rapporteurs. Yeah.